let me introduce Jantina de Vries, who is Associate Professor in Bioethics at the Department of Medicine at the University of Cape Town. Her presentation is being given in collaboration with Andrea Palk, who is a lecturer in the Unit for Bioethics in the Philosophy Department at Stellenbosch University, Western Cape, South Africa. So Jantina will be speaking from a conceptual point of view about the bioethics of polygenic risk scores.
Excellent. Thanks. Let me draw the talk up quickly. So I think you can see the talk, but not yet the presentation. Is that right?
That's great.
Excellent.
Perfect. Okay.
Cool. Excellent. So thank you all for inviting us to present some of our work here. I'm going to switch my video off, if that's okay for you. First to say that this is really Andrea Palk's work. She developed a paper and she's been working on this topic for a while, or she was at the time. But Andrea was unable to make it here today and asked me to present on her behalf. The credit goes to her, and a lot of the work is hers as well. So credit where credit is due, and this is certainly not my work. So thank you for having us. This paper, now quickly, yes, cool, is based on a paper that we collaborated on a couple of years ago, where we were starting to think about the potential uses of clinical polygenic risk scores in psychiatry, under the leadership of a psychiatrist at UCT, Dan Stein, and Andrea as the philosopher in that work. Now, you may or may not have read that paper, and what I'm going to do today is largely kind of give you an overview of the kind of argument that we developed, or the kind of ideas that we developed, but updated from kind of more recent work as well. Now, just to give some context, in late 2018, when we were researching the literature, there were only very few papers which considered the clinical potential of polygenic risk scores, and these are included here at the bottom. And there certainly weren't any papers focused on the ethical implications and challenges that may arise when one would want to use polygenic risk scores clinically. And what we tried to do was an initial exploration, and no more than that, of some of those potential ethical issues, specifically in the context of psychiatry. Now, a focus on these ethical concerns that are elicited by using polygenic risk scores in psychiatry is not a distinct or a novel one, and is situated within, obviously, a larger ELSI work around neuropsychiatric ethics and ethical discussions pertaining to potential biomarkers in psychiatry. So in our assessment or understanding, the discussions of PRSs and their clinical uses is not a unique or novel or completely isolated one, but needs to draw by necessity on many other discussions in bioethics as well. Although we do agree with Catherine Lewis, who published a more recent paper with another colleague, that what is possibly needed are new perspectives on familiar ethical issues. So I think, and that's really important to bear in mind, that the work that is required isn't necessarily completely new, but we need to start thinking about what we've done before in new ways. Now, what I'll do in this presentation very briefly, and especially I'll cut the first bit a bit shorter, because I think Steve did a brilliant job of walking us through the potential for polygenic risk scores. What I'll do is provide a little bit of background information and consider how polygenic risk scores have been spoken about in literature with regard to their clinical use. So I take Steve's point, and I think I agree when he said we ought not want to use these in the clinic, because that's simply not where the science is at the moment. Certainly in literature, there have been speculations about how polygenic risk scores could potentially become of relevance in the clinic. Now, I'll follow that with the main part of the presentation, which is an elaboration of some of the ethical questions and concerns that we identified at the time, although that focus does not aim to be exhaustive. Cool. So we've heard from Steve already that there has been a wonderful development in terms of our understanding both of genetics of psychiatric illness, but also use and limitations of polygenic risk scores. But what has also become apparent in that work is that there is both the enormity and complexity of the tasks at hand, and that this is certainly not smooth sailing. So identifying and using polygenic risk scores in the clinic is certainly not something that is imminent or that maybe ever should be done. Now, whilst genome-wide association studies and whole exome sequencing studies have increased our knowledge of the genetic architecture of psychiatric conditions, most psychiatric disorders, and I know that I'm probably preaching to those who know this much better than I do, most psychiatric disorders are multifactorial and polygenic in nature. And in other words, what that means for those of us maybe like Betty and myself who are not geneticists or knowledgeable in this arena, what that means is that the risk for such disorders is confirmed by the additive effect of thousands of independent genetic variants of small effect and by non-genetic environmental factors and interactions. And in order to robustly identify associations between genetic variants and phenotypes, and thus generate useful genetic risk prediction information, these studies require massive sample sizes, and we know this very well from GWAS and genomics in general. And this has led to a number of meta-analyses that involve the pooling of results from numerous studies across the globe with ever-increasing statistical power. Now, PRSs, as we've heard, are a research tool which is calculating using summary statistics from GWAS discovery samples in which millions of single nucleotide polymorphisms have been scanned to identify those variants that distinguish cases from controls in the particular phenotypic trait or disease that is being studied. And Steve reminded us that what is important there is to be accurate and specific about how phenotypes have been established and what data supports the particular categorization of people in particular kinds of illnesses or conditions. Now, the most common way, well, let me skip that because I think Steve spoke about this quite well, but what is quite important is that the adaptation of polygenic risk scores for clinical use in psychiatry is quite a long way off still, and that there is a real question about whether this will ever, or in what cases, be relevant or useful for patient characterization or classification in real life contexts. Now, various potential benefits of clinical translation have been considered in literature. So Lewis and Vasos wrote one of the first papers on the subject of clinical translation, and they mentioned some of the advantages and benefits that I've listed here. Now, on a practical level, they described that polygenic risk scores would be user-friendly and cost-effective in the sense that the calculation of an individual's polygenic risk scores would be relatively straightforward and require only a single sample of their DNA. So, and because nuclear DNA is supposedly stable from birth or largely stable from birth, the obtaining of a sample could be a once-off, and the calculation of particular polygenic risk scores or generation of GWAS data for a person or whole exome data would be accessible and relatively cost-effective. Now, at a clinical level, receiving genetic risk information could be justified by the benefits of possible preemptive interventions or avoidance of environmental stresses known to trigger onset, thereby enabling possible prevention or mitigation of disorders, such as in the case of schizophrenia, for instance, counseling someone to avoid cannabis usage or other substances as a mitigation or prevention strategy. Now, some other authors, Murray et al., who wrote in 2020, polygenic risk scores should be used or could be used in conjunction with other measures and assessments of risks, including family history or psychometric tools. So in a way, the clinical use suggests that one could use polygenic risk scores to support early detection of patients at risk, possibly in conjunction with other assessments in order to prevent the onset of disease. And at a practical clinical level, polygenic risk scores could be used for busy primary care doctors as a screening tool for patients who are deemed at risk. So one could, for instance, imagine a scenario where all patients presenting with particular phenotypes would be subjected to a genetic test that results in a risk score, and those that are in a particular category at high risk could be then referred to specialist care more timeously. And that could be significant because conditions sometimes, early diagnosis would help in the prevention or preventing the deterioration of conditions. Now, at a public health level, polygenic risk scores could be used to develop intervention thresholds or for resource allocation purposes. Now, of course, currently, as we were reminded quite strongly, the status quo is that polygenic risk scores are not sufficiently robust to be used in clinical contexts. And Steve reminded us about the important issue of missing heritability. So the current status quo is not quite that we can use these things, but literature seems to agree that at least there is tentative support for the potential uses or benefits that may be associated with translating polygenic risk scores into clinical contexts, whilst also being aware of current limitations. So I will now turn to the main focus of this presentation, which is to elaborate some of the ethical concerns that require consideration. But before doing so, however, it's important to consider the ethical justification for disclosing a polygenic risk score in a clinical context. Now, in terms of the dominance, what is the enduring and dominant paradigm in bioethics, which is, of course, principalism, disclosure of a polygenic risk score is justified in terms of a right to receive or have access to personal medical information that may be meaningful or helpful on the grounds of a respect for individual autonomy. The assumption is that individuals do have a preference to freely make decisions and act based on the possession of full and relevant information about themselves. However, where there is a preference not to receive such information, then autonomy also grounds a right not to know or not to receive information if this is the individual's preference. Now, this right to information and the principle of autonomy itself and the concomitant obligation to disclose is not absolute and may be limited if disclosure is associated with a significant risk of harm. In other words, autonomy must be balanced with other goods like avoidance of harm, which may override it. And this is, of course, how paternalism is typically justified in medicine. Now, determining whether disclosure of risk information is ethically justifiable on the traditional grounds of autonomy, therefore requires a full consideration of the risks and benefits of disclosure. In other words, what would be the potential harms of disclosure, or in a wider sense, of using polygenic risk scores in non-research contexts? And how likely are these harms to actualize? What are the benefits of disclosure and wider use of PRS, and how do these outweigh potential harms? Now, in terms of the right to receive relevant and meaningful medical information to inform decision-making, or to opt out of receiving such information, it is worth making two further comments. Currently, when genetic counseling for psychiatric disorders is sought, it would be in cases of a familiar history of a disorder that is highly heritable, such as schizophrenia, or where there is a risk of dominant or recessive inheritance of diseases associated with cognitive or psychiatric impairments or disabilities. Genetic counseling like this does not typically require genetic testing. But the growth of direct-to-consumer genetic testing in the commercial realm has increased interest in the kind of information delivered by such companies, coupled with growing public awareness of the genetic underpinnings of psychiatric disorders, which will likely lead to increased uptake of genetic counseling. Now, in addition, and as we pointed out in our paper, studies indicate that should genetic testing for psychiatric disorders become possible or available, there would be significant uptake. And here it is worth mentioning that DTC genetic testing companies already offer polygenic risk scores for hundreds of diseases, disorders, and traits. And this information is provided without genetic counseling. And what is particularly interesting is that Murray et al. In their 2020 review mention that of the more than 28,000 people who have uploaded their DNA data that they received from a DTC genetic testing company into the free online tool ImputeMe in order to generate their personal polygenic risk score for various traits, the second most actively searched for condition after obesity was schizophrenia. So there appears to be an indication that there is a preference for or an interest in receiving this kind of result. So people want this, seem to want to, or at least those 28,000 people wanted polygenic risk score information on schizophrenia. So regardless of whether we think or say that there is no clinical use for this, or that this is actually still a research tool, this information is being accessed by the public in ways that, we must discuss whether that is appropriate and whether the evidence that supports the giving out of those risk scores is robust and relevant. Now, possibly the most urgent ethical concerns regarding the clinical translation of polygenic risk scores is the potential to exacerbate health inequities. And this is, this point overlaps with what Steve was also saying. So in addition to the diversity gap that has been tracked several times, we also need to think about whether and to what extent the pursuit of or the development of polygenic risk scores and their clinical translation would or could enhance global health inequalities. Now, in terms of the diversity gap, we know that there are initiatives underway, including several that are run from the Stanley Center that specifically seek to address the diversity problem, including studies on the development of neurodevelopmental disorders in South Africa and Kenya, for instance, but also many different studies, including some psychiatric studies under the Age Through Africa Consortium. Now, in relation to the health equity point, whilst obviously a concern around global health inequities or health inequities, even in national contexts, intersects with the diversity gap, it's not the same. And I hope that I don't need to belabor that point, and I hope you'd agree with me. Now, for instance, in Cape Town, risk factors for psychiatric illness and the epidemiology of some psychiatric illnesses importantly overlap with poverty, which exposes people to many of the factors that trigger conditions like schizophrenia, including things like surviving violence and substance abuse. And I don't know how well you know South Africa or whether you know South Africa at all, but there are many people in this country who do not have effective access to healthcare and who live in informal settlements around our big cities in situations where they have colliding epidemics of violence, including gender-based violence and femicide, in conjunction with a high burden of HIV and multi-drug resistant TB, and so on and so forth. Now, against that background, it is not clear whether and to what extent having high polygenic risk scores for psychiatric conditions in certain patients does anything to help protect those persons against the structural social conditions that increase their risk. Many people are trapped in poverty and cannot escape where they live. And so it is not clear to me to what extent that changes anything about how a person or a medical doctor changes recommendations for what that person should avoid, for instance. So those concerns, I think, are really important in the context of polygenic risk scores, and we can speak about that more. Now, another really important concern, and one that is getting increasing attention in the literature, relates to the potential for stigma or harm, potential for discrimination, and the abuse of polygenic risk scores to further discriminate against certain groups of people. Now, we know, and Paul's group and others and several people on this call, I think, have done work on the relation between genetics and stigma, and we increasingly know that the picture isn't as straightforward. But what is really important is that there is a possibility for us to use polygenic risk scores to predict things that we perhaps think shouldn't be predicted in that way. So, you know, this is a snapshot on this slide of a difference of opinions about the use of polygenic risk scores to predict non-heterosexual behavior on the African continent. Same-sex marriages and sex acts are illegal, and people could end up in jail. And so you could imagine a potential really contrary use of polygenic risk scores to predict and to further categorize and stigmatize people. Now, just to point out that I don't think that issue is necessarily completely different from a lot of the discussions we've had in the context of dual use issues. You know, I think that there's a lot that we can learn from those dual use discussions to think about this issue further. But just to point out that I think it is this is a real problem or a real challenge to the use of the social use of polygenic risk scores. Now, sorry, let me just. So related to this then is the potential of the interpretation of polygenic risk scores to exacerbate stigma, as I've already said, and just to point out that whilst we have concerns about how genetic information could be used to increase or decrease stigma, we also have very interesting work by Celeste Condit and some of our own work in Cape Town that really points to the fact that people are strategic essentialists, which I think is important in the context of polygenic risk scores, because it helps us think through whether and what the nature of the harm is that we worry about. Now, one real risk, and that is one that Lewis and Green report in their paper as well, is a question about whether the communication about polygenic risk scores in a clinical context could lead to either an underestimation of roles played by social environmental factors, but also to promoting and fostering kinds of behavior that are both in patients and clinicians that are just not helpful in how we think about or treat psychiatric conditions. Now, the third area of concern discussed in our paper and also raised in a more recent paper by Doherty et al. In 2021 is the potential use of polygenic risk scores for prenatal testing or the testing of minors. And in terms of the testing of minors, there is some justification in cases where there is, or some apparent justification, in cases where there is a family history of a disorder, particularly those disorders for which there are certain avoidable risk factors that increase vulnerability, such as, for instance, cannabis or stimulant use in adolescence. Now, there has been ample engagement in the literature on the ethical permissibility of genetic testing of minors. And so again, that is not an issue that is entirely novel. But testing for polygenic risk scores is likely to intensify concerns about their use in minors, particularly given that the disorders for which polygenic risk scores would be used to predict risk would likely to be those with the highest heritability, such as schizophrenia and bipolar disorder, both of which are subject to high levels of stigma. And in particular, there would be a high risk for internalized stigma and negative self-labeling in such cases. The other potential use of polygenic risk scores would be for various forms of prenatal tests, including pre-implantation genetic testing. And I think Steve was going to comment on that, but jumped it. Now, what is concerning there, and this is a point that has been kind of pointed out by several authors, is the reintroduction of a eugenics kind of orientation to genetics, which selects for some desirable people and not others. And the concern is validated by the fact that polygenic risk scores are indeed being marketed in the commercial realm for such purposes, including the possibility of testing embryos for low IQ. Now, polygenic risk scores could also be used to test for mental disorders, this is already, as the headlines at the bottom indicate, this is already a topic of discussion. Now, obviously, and I've pointed previously to the overlap with other ethical debates, and there are important overlaps between these concerns and those that have been articulated in the context of germline genome editing, as well as in the context of commercialization of stem cell or alleged stem cell therapies and treatments. Now, in the context of germline genome editing, the need for regulation has been repeatedly emphasized, as has the need for what some have called broad societal debate. And both of these probably need to be emphasized for the use of polygenic risk scores as well. So regulation and societal debate about the desirability of some of these application of polygenic risk scores seems to be really important. Now, the final concern engages with one of the most likely applications of clinical polygenic risk scores. And those are the cases in which a consenting adult has submitted to PRS testing for screening purposes, and in particular challenges associated with feedback of high polygenic risk scores for developing psychotic disorders such as schizophrenia and bipolar disorder. Now, this concern is situated in the broader domain of the ethical challenges associated with feedback of genetic findings, and issues such as privacy and confidentiality, implications for family members, and appropriate ways to communicate such information are relevant in terms of the last point about potential psychological distress. Now, this is particularly important given findings reported by Peck et al. In 2022, who conducted a study to ascertain why individuals used direct-to-consumer genetic testing data to generate polygenic risk scores. And what they found was more than half of the participants experienced a negative reaction after receiving the polygenic risk scores. And 5% scored over the threshold for potential PTSD disorders. So of those people who wanted to or who used DTC testing to generate polygenic risk scores, just to emphasize that point, a very large number had negative reactions, and many or most, and 5% had scored over the threshold for PTSD. Now, lower understanding about polygenic risk scores in that study was associated with experiencing a negative psychosocial reactions. And those kinds of findings are extremely important for the topic at hand. However, what we focused on in this is a more recent paper, but what we focused on in this final section of our paper was the potential difficulties associated with communication and interpretation of results, given the etiological complexity of psychiatric disorders and challenges pertaining to the understanding of complex risk information. Now, studies indicate that risk tends to be interpreted according to a subjective framework with considerable variation between subjective estimates of risk and so-called objectives for statistical risk. And studies also indicate difficulties in the interpretation and understanding of genetic information, in particular complex genetic interactions. Now, the comprehension of polygenic risk thus represents an intersection between various constructs that are difficult to communicate and easily misinterpreted. And an added complication, which I have already touched on, pertains to the tendency for genetic information to be interpreted in ways that we don't always understand and that could be reductive or deterministic. And the risk of this is high because while overall risk of disease can be reduced through interventions, genetic risk itself remains relatively stable. And it would therefore be important to ensure that communication of a high polygenic risk score does not support such interpretations that are deterministic, thereby leading to prognostic pessimism or fatalism, whereby the role played by non-genetic factors in health is discounted. And ensuring adequate understanding would be crucial because the aim of communicating a high PRS for a psychiatric disorder would be to ensure that any therapeutic recommendations or preventative measures are heeded. Now, by way of conclusion, a few suggestions. One is that if PRSs are ever utilized in clinical context, then research regarding effective communication would be essential as a means of ensuring constructive interpretation. And that, moreover, given the shortage of genetic counselors, at least in our context, South Africa is better off than the rest of the continent, but even in our country, we hardly have any genetic counselors, like literally 2 hands full, and that's it. And therefore, it would be likely that healthcare professionals who do not have expertise in genetics would be the ones to feed back such findings. And it's obvious that because of potential harm relating or misunderstanding relating to the return of these results, it's very important that there is appropriate training for staff that is required to feed these back. And then finally, it would be necessary to consider how PRS findings could be translated into more accessible formats whilst ensuring that it doesn't lead to oversimplifications or reductive interpretations. And then tools that assess genetic literacy and the comprehension of risk information could be adapted for this purpose. So that is the full extent of what I wanted to share with you today.
Thank you, Jantina.
Excellent.
That was such a beautiful way to walk us through these issues. I really appreciated that. Steve, I see you've unmuted yourself, and
yeah, I just wanted to say that was a beautiful talk, and also thank your colleague who couldn't come. It was very, very balanced and thoughtful. I'm one point that has always weighed on me, and I have to say, since I was NIMH director back, I can't remember, 1 or 2 centuries ago, but is this issue of what you describe people who are exposed to overwhelming environmental factors. You know, we can show a correlation between poverty and its correlates and risk, but the issue is what to do about it. It becomes, it's almost like changing your genome, and societies, I learned the hard way that when you discuss this with policymakers, they said, well, we already know poverty is bad, next. So I wonder, I mean, I do wonder about whether it makes any sense. This is partly a technical question, partly a policy and ethical question, to if studies well enough power to look at people who develop these conditions despite having a less loaded, less severe polygenic score, and conversely to look at resilience. And I wonder if there's a way of sorting out and we may discover that what's underneath poverty is very much analogous to polygenic disease, right? So many small insults that we can't figure out what to do with it. But I just wonder whether people have an appetite to begin to turn these very large, what I've called proxies, into factors that we might be able to intervene in, right? Maybe there are some, I mean, you mentioned cannabis, as difficult as it is to prevent cannabis smoking when people are distressed. But, you know, do you think that would be a feasible, you know, and ethical approach something that could happen at UCT or elsewhere?
So the declaration is I'm not a scientist and I'm not a psychiatrist either. And so, I mean, my response would, nor am I South African, nor am I South African Black. And so, you know, I mean, I speak from a particular viewpoint that is probably ill-informed, but, or at least partially, is very partial. I think much more work, you know, needs to be done to think about these complexities, and perhaps it's appropriate to do that in the global South, right?
Yes.
I don't think these complexities are unique to our situation, and I think this is partly about diversity, right? It is ensuring, you know, I mean, we know this, and there's increasing evidence that diversity is not just about, you know, the people that we include in genomic studies, i.e. the research participants, but that diversity also matters in terms of diverse workforces, for instance, leadership, high-level leadership of scientific projects, having researchers from very diverse contexts who have access to different ideas about what matters in how in disease epidemiology, for instance, changes how we articulate particular scientific questions. And so, I mean, that's a broad answer, but I think that's...
No, I think it's a good answer.
Yeah.
Yeah.
What I want to say, I mean, if I just look at the South African context, there's 35% overall unemployment, but in the informal settlements, that can be as high as 50 or 60%. Now, young people living in really crowded informal housing shacks, where gangsterism is rife. Cape Town has the dubious honor of being the murder capital of the world. That doesn't happen in my neighborhood, right? People get murdered in areas that are crowded, where there's 50, 60, 70% unemployment, where your only option is to join a gang. If you're a woman, you know, chance of you being murdered, femicide is through the roof, rape is dire. So you know, what does polygenic risk scores, I mean, how does that change, right? It changes nothing for somebody who takes a taxi back and can't change where they live or what they're exposed to. And so, I mean, I think it's a really important question, is how do we accommodate that reality for how we think about global health equality, for instance, right? We recognize it's important, but how do we incorporate that substantively into the genomics work that we do in psychiatry and other areas?
Thank you. Dr. Lebowitz.
Yeah, thank you so much. And thank you, Jantina, for that really excellent talk. I thought the breadth and depth were very impressive, and you touched on a lot of really interesting issues. As someone who's particularly interested in many of the things you talked about, but especially in how people react to or make sense of genetic information, I really liked what you had to say about the potential risks and benefits of disclosing polygenic risk information to individuals. And I noticed that you focused a lot on the risks of disclosing high PRS, such as how that might lead to prognostic pessimism and other downsides like that, which is certainly something I've thought a lot about and think is very important to consider. But I also wonder about the perhaps less readily appreciated risks of disclosing low risk information, low PRS or low genetic risk information that may still be worth considering, particularly in terms of, for example, false reassurance. You talked about the potential for PRS to be used in primary care to screen, to, you know, I guess, separate people into those who are higher or lower risk. So what happens to people who PRS suggests are lower risk, but actually have other risk factors, and maybe they or their clinicians might downplay the risk, and or their treatment providers or healthcare providers, and what could be the negative consequences of that. It's of course related to something you did touch on, which is the risk more generally of downplaying non-genetic factors as we focus on PRS and other genetic measures, but not exactly the same. I just wondered if that was something you thought about at all.
So the short answer is no. But I agree with you that is very important, particularly in the relation of commercial companies sharing these kinds of low risk PRSs, and that is not, it doesn't just happen in high income countries. In South Africa, we have a burgeoning commercial environment where, dietitians and all kinds of people feedback low-level risk scores for all kinds of things. So I agree with you that is really important. We haven't really kind of done much work on that yet, but I agree that should be done. But I did realize I didn't put the final slide on, which has the references. I wonder if I should place them in the chat, because there are some papers that or I can send you the.
You could do that, yeah, because I actually wanted to follow up on one of the papers that you discussed, and I wasn't aware of it, that had talked about, adverse impacts of communicating polygenic risk scores. Because I've looked for papers like that and have been unable to find them. So
yeah, so that was a recent paper in the European. Sorry, I can't for some reason seem to paste it in the chat. Let me quickly show you I'll upload the slides, share the slides again, so that you can see that final slide. Yeah, so that's where they are. Whoopsie, sorry about that.
great.
Cool. So that was a recent paper, and I think those findings are really interesting, right? 60% of people reporting negative reactions, and 5% of those classify or potentially could be diagnosed with PTSD. I not a psychiatrist, I'm not sure how robust that study was, but I think that's a really interesting finding.
Yes. Which paper is it?
It's the final one, PIC, the 2021
Oh, okay.
European Journal of Human Genetics.
Thank you.
It would be great to have those in the chat.
Yeah, I've tried to.
We will also be posting the recording of this session on our website. And so you'll be able to see that slide when you look at the recording, but great. If you don't mind posting it in the chat as well, that'd be wonderful, Jantina.
Cool. I'll try to do that. That's wonderful. I'll proceed to do that.
Okay, thank you.
As we